Alright, so this one is about DNA fingerprinting and DNA profiling, which are actually two different things. We'll start with DNA fingerprinting, which was developed in the mid-80s down the road in Leicester. And the idea is this: if we took a section of DNA, I'm just going to write out a more or less sort of random thing here. There's all our bases, so if I paired those up, we'd have something like that. Okay, there's my two strands of DNA, and they're all paired up nicely. Now, we probably get the idea that this DNA codes for a gene. Okay, this might be a gene to make a protein, a particular enzyme, a hormone, whatever it may be. But it turns out when you look at DNA, actually huge sections of it, apart from the bits that actually do the coding for genes, there are large sections of it that don't. And when you look at these sections, they're very often just a repeating sequence. So maybe it's just T, A, T, A, that's quite a common one, over and over again. So they kind of, what you get on a chromosome is a region of DNA that gives you a gene, and then one of these regions where it doesn't really do anything, and then another bit of a gene, and then another region where it doesn't do anything. Okay, and these regions are called satellite or mini satellite stretches of DNA. Okay, now what was discovered in Leicester was, if you compare two people's DNA, our DNA is fairly similar, our genes are fairly similar. So I've got a gene, for example, that would make insulin. If you had that gene, it would pretty much be the same. It would have the same sequence of DNA as mine does. But these satellite sequences can be different, and they can be different lengths. So this could go on for a longer section in somebody than in one person than somebody else. And you maybe get little bits of variation in here as well. Now, between two closely related people, these mini satellite sequences are actually quite similar. But again, it turns out they're actually unique for every individual. If you're wondering what these do, by the way, it's not quite clear. Nobody's quite sure yet. We used to think that they didn't do a whole lot. They used to be called junk DNA. One thing it might do is it cuts down the problems of mutation. Imagine if we changed one letter in here, all the rest of the DNA would kind of shuffle along. If we took those two letters out, for example, all the rest of the DNA would get shoved along, and that would alter every single gene. And it's thought maybe that these satellite sequences help to stop that by sort of cutting down this problem. It ends up that each gene further down the line it isn't affected by an early mutation, if that makes sense. But that's getting a bit sort of beyond what we need. What we need to think at the moment then is we have these mini satellite sequences. And what you would do, you had to get a fairly decent sample of this. So you would need, perhaps a good spot of blood or saliva, and you need quite a bit of it. And what they'd do is they'd take the DNA and they would cut it up using enzymes. And these are special type of enzymes. They're called restriction enzymes. And what they do is they cut up the DNA into smaller fragments. Okay. And what we're really interested in is these mini satellite sequences, because they're the bits that are going to show a difference between individuals. Now, what we would get then is these satellite sequences of DNA, these bits that have been chopped up, broken down. What you would do with it is you can split that DNA up, you can heat it, okay, and it splits it apart. And what the team in Leicester did is they developed something called a DNA probe. And a DNA probe is a section of DNA, but it's a single strand. So for example, if I was looking for this section of DNA here, I'd want a complementary strand to it, so I would do something like that. Okay, and I stick these probes in, and if it comes across a section of DNA that it matches up to, it will join up. Okay. And why do we bother with these DNA probes? Well, we can make them different lengths, and we can use it to identify these mini satellite sequences. You then put your attached strand with your DNA probe in it, and you use a technique called gel electrophoresis. And what this does, it's basically you put your DNA sample into a little gap, and this is kind of jelly stuff, and you put it in a machine, and at one side you've got a positive charge, at the other side a negative charge, and this would be kind of this would have a liquid in it that would allow electricity conduct. And basically what happens is these DNA, these little sections would spread out, okay, and they get attracted depending on what kind of charge they've got. And you end up with a series of bands, and this is the kind of thing you maybe see it on films and things where they have these different bands. So if I knew that was a sample, for example, from a murder scene, and I tested somebody else's DNA and it came up like this, I might be able to say, it's a different pattern. And if I tested another one and it came up like this, I could look at it and say, oh goodness me, that matches my fingerprint over here, and I've got a match. How accurate was it? It's not 100% accurate, but you're looking at somebody matching DNA. You're looking at something like perhaps in a billion, 1 in 2 or 3 billion chance. So it's not 100%, but it's getting pretty secure. And the problem with DNA fingerprinting is you need a good sample, so you need quite a bit of stuff. And what was happening with crime scenes is we were finding that you didn't always get a good sample. You maybe only get a tiny amount of stuff. So rather than finding a hair, you maybe only find a few cells, much more difficult. So DNA profiling is a more advanced method, really. And what you would use here is if you've got very small samples, perhaps just 1 or 2 cells, you would go through this same process. You'd break your DNA up, you'd put the DNA probes in. But we also have a process called PCR, or polymerase chain reaction. And what this does is, once you've got your DNA copies here with the probe on, it makes lots and lots of copies of it. So rather than just having a few, you end up with lots of them. Why is this important? Because in order to do the DNA fingerprinting, you need a lot of these sections of DNA. So really this PCR magnifies, if you like, it amplifies the amount of DNA you've got. The problem with it is if you contaminate your sample, so if somebody was getting a sample from a crime scene and some of their own cells got into the sample, their own cells might also be copied. And so their DNA would show up. So people who work in crime labs would all have to be DNA tested beforehand. They'd have to have a record of their DNA in case it showed up in a crime scene, and you'd know then that it had been contaminated. And another thing they can do, which is a bit better, is these DNA probes can have a chemical attached that will fluoresce, and fluoresce means it will give off light. So when it attaches, when the DNA probe finds something it matches, a good word in here, it's complementary. A complementary strand will match up. A is complementary to T, C is complementary to G, for example. They always match up. If it matches up, it would give off light. And so when you look at these kind of pictures, they often sort of look as if they're glowing a bit. And what you would do is you'd put that through a computer, rather than sitting there and counting them like I did before. You'd put it in a computer, which could very quickly work out if there were similar DNA profiles of people or not.